Hello everyone, I am Shruti Parashar. I welcome you to my channel Biology 360. Today we are going to discuss structure of anther and microsporogenesis. This topic is helpful for ICSE, CBSE, IGCSE and HSC board students and for students preparing for NEET, AIMS and Olympiad exams. A typical stamen has the long and slender stalk called the filament and a terminal bilobed structure called the anther. So in this diagram we can see a long stalk, a slender stalk-like structure which is called as filament. So a typical stamen has a slender stalk which is called as a filament and a terminal bilobed structure. So this is a bilobed structure that we see in the picture called as anther. A typical angiosperm anther is bilobed, with each lobe having two theca, that is, they are dithecous. So this is the 3D section of anther. In the anther, there are pollen sacs, and in the pollen sacs, pollen grains are formed. In this slide, we see a transverse section of a young anther. A young anther has connective tissue, epidermis, endothecium, sporogenous tissue. And then in the diagram B, there is an enlarged view of one microsporangium showing wall layers. So each microsporangium, it has different layers such as epidermis, endothecium, middle layers. Then there are microspore mother cells which help in the formation of microspore or pollen grains. And there is another tissue called tapetum. And see picture, it shows a mature dehisced anther. Transverse section of an anther. The anther is a four-sided, that is tetragonal structure consisting of four microsporangia located at the corners, two in each lobe. So in each lobe there are two microsporangia. So there are total four microsporangia in the anther. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grains. So inside the pollen sacs, pollen grain formation occurs. Structure of microsporangium. A typical microsporangium appears near circular in outline. It is generally surrounded by four wall layers. So a microsporangium has four wall layers, namely the epidermis, the endothecium, the middle layer and the tapetum. And each of these layer, it performs some or the other important function. The outer three wall layers perform the function of protection and help in dehiscence of anther to release the pollen. It means the epidermis, endothecium and middle layer, they give protection and they also help in dehiscence of anther to release the pollen grains. The innermost wall layer is the tapetum. It nourishes the developing pollen grains. So we can say that tapetum is a nourishing tissue or it's a nourishing wall layer. Cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. When the anther is young, a group of compactly arranged homogeneous cells called sporogenous tissue occupies the center of each microsporangium. So in the center of each microsporangium, we see a compact tissue called as sporogenous tissue. Microsporogenesis. Microsporogenesis means it's a process in which there is formation of microspore. Genesis meaning generation or formation. formation of microspores. As the anther develops, the cells of the sporogenous tissue undergo meiotic division to form microspore tetrads. As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or microspore mother cell. So sporogenous tissue, it undergoes meiotic division, yes, and it forms microspore tetrads. And inside the microspore tetrad, there are microspore mother cell that helps in the formation of microspore or pollen grains. The process of formation of microspores from a pollen mother cell, PMC, through meiosis is called microsporogenesis. The microspores, as they are formed, are arranged in a cluster of 4 cells called as the microspore tetrad. As the anthers mature and dehydrate, the microspore dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspore or pollen grains are formed that are released with the dehiscence of anther. So, anther, it undergoes dehiscence, and due to which large number of microspores or pollen grains are released. And these pollen grains, they help in sexual reproduction in angiosperm. Friends, do subscribe to my channel to get all the latest videos on biology. Thank you.